Well, thank you again. Thank you for joining. So today we are going to explain more about what the FDCube can do for you. My name is Peter-Bram Hoen. I'm professor of bioinformatics at the Radboud University Medical Center and also developer of fair data infrastructure. And we are presenting also on behalf of the X-omics Consortium, a national infrastructure for the generation and the analysis of multi-omics data. The details of the FDCube will be explained by Firdaus and Xiaofeng later. Instead, what I'm going to do to you is discuss with you the challenges that we face in multi-omics data analysis and how fair data technology can actually overcome some of the hurdles that we know in multi-omics data analysis. Before I do that, I would like to see your responses to the next question, and that's actually asking you to indicate whether you are already analyzing multi-omics data yourself, or whether you're still analyzing a lot of single omics data, or whether you are not experienced in the analysis of omics data at all. Maybe you can 18 responses so far, and the majority of you are analyzing single omics data. Some have experience with multi-omics data, and some actually still need to start their analysis of omics data, or are maybe interested in the FDCube infrastructure because it also may facilitate other types of analysis than omics analysis. Thank you for these answers. What is the FDCube about? The FDCube may help you to answer complex questions related to a specific biological problem efficiently and correctly by accessing information in multiple sources automatically. That's also where we see that fair technology has the biggest impact, and that is when there is multiple sources of information from which you want to integrate data. So when all these data sources are fair, this integration should be facilitated. And in the example that Firdaus will demonstrate later to you, we were, for example, interested in the pathways that were affected in COVID-19 and which would be supported by both transcriptomics, proteomics, and metabolomics data that is gathered from COVID-19 patients. And from that, we needed, first of all, access to the omics data themselves, but also access to the pathway information that records which transcripts, proteins, and metabolites are involved in a certain pathway. So this example will show to you how a complex question can be answered using data from multiple sources that are all fair and therefore more easily integrated. So the FAIR principles were already defined in 2016 or a little bit before, but in 2016, the FAIR principles were first published in Scientific Data. And I'm sure that you have seen this paper, even cited this paper, because it's cited around 15,000 times already, I think, at the moment. And it involves 15 guiding principles. But what I would like to highlight on this particular slide is that already in the abstract, it says that it's not just about making data accessible, but also interpretable by machines. And well, increasingly, we see that as important also with the evolving AI technologies. So we may actually now also refer to the FAIR principles as fully AI ready data. And I hope that we will be able to illustrate the advantages of that to you today. So data and also services should be fair for both humans and machines. What does that mean in the context of omics data? First and foremost, I think it should be the case that the data sets that you would like to use for your research, the published data sets, should be findable. And that also the data should be accessible with a clearly defined access protocol. Moreover, what I will show later is that the data that is currently out there is hardly ever interoperable. And the use of vocabularies, standardized vocabularies and ontologies, for example, for identifiers of genes and proteins and metabolites, are still underused and not accessible in a machine accessible format. And this all hampers the reusability of published omics data. In order to be reusable, we need to add more contextual information, allowing the proper interpretation of the published data, and also need rich provenance information that facilitates also the citation of the data and giving credits to the producers of those data. There are a number of misconceptions that I often hear around FAIR. And in the context of multi-omics data, this one is one that I hear a lot. The data already in the European Genome Phenome Archive or a related database from the NCBI, the EGA is from EBI, or the proteomics database at EBI, PRIDE, or the metabolomics database at EBI, metabolites, and they are therefore fair. Well, that's only partly true. So most of the data in these repositories is indeed findable and also accessible through an access protocol, but hardly ever interoperable. And making data interoperable is probably also the key to reuse of the data. So they are there, but the description of actually the meaning of the data elements, the metadata around the data sets is rather poor, and that prevents the reuse of data. Another misconception that I often hear is I cannot make my data fair because they are sensitive. And there is a kind of a difference between like putting data in the open and making data fair. That is really not one-to-one related. Fair data can also mean that you actually make the data available on the very strict agreements with data use agreements, etc. They do not need to be open. The metadata, so the data describing the data, should be open. In that metadata makes it findable and interpretable, but the actual access to data can be regulated and does not need to be completely public. A third misconception around FAIR is that making data fair is a huge effort with little return on investment. And I would say this is partly true. I think making omics data fair is still a significant effort. With the FDCube, we try to make your lives a little bit easier, but it's still an effort. The return on investment is actually quite big, because what I've seen in my experience, when you make your data available in a format that is easy to use by other researchers, it will actually lead to reuse of your data, to increase citations and visibility in the scientific community. So in the CrossOmics Consortium, so the Dutch infrastructure for analysis and generation of multi-omics data, we really focus on the enabling of efficient data integration with both data generated within the CrossOmics facilities, but also with external data sets. CrossOmics has adopted the FAIR principles and takes them a step further by helping researchers to make their data analysis workflows transparent and reproducible. So what do? We try to make your research fair at many different levels. So we stimulate the collection of fair metadata on samples, and that is actually illustrated also today. We make sure that you collect sufficient information on the protocols that are used for data generation. The FDCube will also help you using identifiers that are uniquely resolvable and therefore unambiguous. And in addition to that, so more on the data generation part, there is also FAIR in the computational part, so the downstream analysis. So we stimulate the collection of FAIR metadata on analysis algorithms and tools that are used, but we also create FAIR workflows. And I have some examples on how we did that in the following slides. In the last point, CrossOmics also deposits their tools and functionalities in the Iatris Multi-Omics toolbox, which I also will highlight in the subsequent slides. So this is a paper that Casper de Visser in the group and also affiliated with the CrossOmics Consortium wrote, and which is available through the link here. We will, of course, provide you with all the slides later on. And it shows you 10 quick tips on how you can also build FAIR workflows. So going all the way from the data to the final results. And some of the FAIR principles, findability, accessibility, interoperability, and reusability also applies to these workflows. And in this paper, you can find hints on how to make also the analysis workflows FAIR. In the action study, that was also a showcase study of the CrossOmics Consortium, which is published in GigaScience here, we also made an effort to make the analysis workflows FAIR together with the data. So we created a FAIR digital object containing the FAIR multi-omics data plus the analysis workflow. And when you would like to read more about this, I can refer you to the paper here. And like I said, we collaborate a lot with the European infrastructure for personalized medicine, and that's called Iatris. And they make, through their website, motbx.iatris.eu, they make all these tools, data, protocols available for you to navigate and to get more support in the analysis of multi-omics data. And most of the tools that we developed within CrossOmics can be found here in the data management and stewardship section of this multi-omics toolbox. Before we go to the common challenges that people may face in multi-omics toolbox, I would like to launch another poll actually asking you for what your major bottlenecks or major struggles are when analyzing multi-omics data. We do that in a word cloud, and I hope when I launch it now that you can write a few keywords showing actually which struggles you are facing when analyzing multi-omics data. There are many things coming up already. 10 people already were able to submit responses. I'm waiting a bit more for others to contribute. Many people are struggling with the data themselves. I'm not sure what they actually mean with that. But the second highest is, I think, the use of different identifiers. Well, we will show in the showcase from Firdaus how we actually handle the different identifiers that are being used by the community. Then there's also issues around the big data and hardware actually that is needed. And well, we have a connection with the Serve Research Cloud that Xiaofeng will comment on, that we will illustrate in the second part of this webinar. And there are several others, like metadata standardization, biological interpretation, incomplete ontologies. I think they are all very valid. Some of them cannot be tackled by FDCube per se, but there's more efforts in the community to standardize well the way how we annotate multi-omics data, and we are engaged with the European efforts there, and others well maybe served by the FDCube. And after this webinar, you're able to judge yourself how useful the FDCube may be for to tackle some of these struggles that you're working with. Okay, so thank you for your answers again. And we continue with the presentation. And well, what I, in my research, found were major struggles is presented on this particular slide. So I think typically a bioinformatician or a scientist well, spends a lot of time on preparing the data and has then little time left for the real data analysis and interpretation. And I think fair data technologies like the FDCube is really geared towards tackling this specific challenge. So a lot of time is spent on reformatting the data, making sure that the identifiers match, etc. So we will tackle this. Then connecting omics layers through known biological relationships that may be present in several databases, but may be difficult to pull into your analysis or at least labor intensive is another challenge that I face a lot and that we hope to well, mitigate at least a little bit through the FDCube. And then, yeah, this is a bit related. So all these differences in connecting data sets, your own data set with published omics data sets is difficult because of the lack of detailed metadata. So the FDCube helps both the data producer to verify the multi-omics data set, to equip the data set with rich metadata, and to increase the exposure of your published data sets. And it also helps the data consumer to analyze, integrate, and interpret multi-omics data. So there is a data producer and a data consumer side that we try to facilitate. Well, why the need for this rich metadata is, I think, illustrated in an example that we ran recently as another cross-omics showcase. And that was a very complex experiment where we actually did multi-omics profiling, so lipidomics, metabolomics, proteomics, whole exome sequencing, RNA sequencing, and DNA methylation on a large set of induced pluripotent stem cell lines from different individuals, so both healthy individuals, but also diseased individuals suffering from a rare disease. And the design of the experiment was rather complicated. So we had both biological replicates and technical replicates. And during the analysis, actually, we found out that although we made an effort to collect as much as possible metadata in the beginning, we still lacked some essential information. For example, we initially forgot recording of the metadata around the passage number of the cells, on the exact disease-causing variants in the proper gene variant nomenclature. We missed some details around the sample preparation protocols and also in the batches in which experiments were run. And that's obviously also very important to take into account in the analysis. So even though we had some initial metadata collected already at the beginning of the study, we noticed that it was important to even enrich the metadata when we were busy with the analysis. And actually, this is also what we try to prevent and make sure that we collect most metadata already at the start. And for that, the FDCube contains a system to capture all of this metadata in a simple Excel sheet that is then transformed to FAIR data. And we collaborate in this effort together with the people in Nijmegen, mentioned here on the screen, working on the FAIR data station. So this is all summarized in the paper about the FDCube, which you can of course read, but will also be illustrated to you by the next two speakers, so Xiaofeng and Firdaus. But before that, yeah, it's possible to ask any questions about what I presented to you today. I'm not sure whether there are already questions in the Q&A. Type your questions there in the Q&A. See question coming in. a question here from Pirjanka. And that is, why did you use a Surf Cloud and not Mydre for the Fair Data Cube? I think that question is best answered after Xiaofeng's presentation. But
yeah,
let's,
that up.
yeah, that we will definitely need to come back to this question. Okay, if any other questions arise, either now or during the next presentations, write them in the Q&A and we will get back to them. And then we now continue with Xiaofeng's presentation.
Okay, thank you. Then I will follow on, and I will share my screen. Oh, sorry, I should change it. Can you see my screen?
Yeah, it's not.
Yes. Yes, but not in the presentation. Yeah.
Yeah. So let me turn on the pointer. Okay. So it's better. Okay. So thanks for Peter-Bram's presentation, which mentioned some of the needs and the benefits, and I will follow on that and present the Fair Data Cube, the main components and interaction between them. So it's more technical on the data infrastructure perspective. And yeah, first I'm Xiaofeng Liao, and currently I'm working in Health AI, and previously I worked in Cross-Omics Initiative and as a data steward and metadata specialist. So Fair Data Cube, we start with a definition. Fair Data Cube is a set of tools and services that helps researchers, in our case for this moment, in omics, multi-omics researchers, in different stage of their research data life cycle, including creating and describing new data, and finding, understanding, and reusing existing fair multi-omics data. We will see these tools and how they interact later on. I would present you with this high-level architecture of the Fair Data Cube. So we see different stakeholders on this diagram. We see researchers, and we can call it data producer or data owner. And on the left, we see data set consumer. Also, they can be researchers. And we have different researchers at a different level in terms of their need of the data and the data set. So from the data set producer perspective, here we can see that Fair Data Cube can help dataset owner. If we look at this arrow, the blue one, it acquires data in different format, and also can describe the data linked to data source, and also can help to deposit the data, and can transfer data, metadata into RDF, and then published in metadata registry. In our case, we use Fair Data Point as this registry and also a data portal. And here I can quickly pick up Priyanka's question, why Fair Data Cube. If I remember correctly, your question is regarding DRE. So here, DRE could be here, external omics data repository, which could be EBI, and also be a private data storage for a digital research environment. And Surf Research Cloud, we will see it helps at the bottom to help researcher to deliver algorithm. But now we look at on the left how Fair Data Cube helps the dataset consumer or a researcher in two folds. The first fold is that the researcher can perform semantic search or query on the metadata portal. And if the researcher finds an interesting data set and would like to go a step further, then in that case, the Fair Data Cube here at the bottom part, the Fair Data Cube has a component called Vantage6, can help the researcher to deliver their algorithm to the data set. The data set stays or under the administration of the dataset owner, stayed in maybe a private data storage, for example, a DRE, and also could be in other external repository, for example, EBI, or even in a complete public one, for example, in GitHub. So that's the high-level architecture of the Fair Data Cube. Here we mentioned several stakeholders and several components. We will continue to see how they interact. But before that, let me briefly mention some of the main components in case that maybe some audience are not familiar with the terms or names. So the first thing I've mentioned is a fed point. It's a REST API and a protocol, and consists of web client. Here on the right, you can see it's a component of fed point. The main function is to create, store, and serve fair metadata. Here we can see a fair data point web client, and in the core is fair data point server. And behind it, there are two storage. On left is the triple store, which is used to store the RDF data, that is the verified metadata of the dataset. And in our case, we're using GraphDB as the triple store, and also fair data point using MongoDB for storing some of administration information, for example, user account, etc. So this is the first concept. Then also at the bottom, with these links, you can get more information about the fair data point on the Read the Doc website. Also provide a very short video on YouTube, which was presented by Luis, where you can quickly know what the fair data point is. Then the next concept or term I would like to mention is Vantage6, which we used as a component in Fair Data Cube. Vantage6 is an open source, privacy preserving federated learning infrastructure for secure insight exchange. So you can see that Vantage6 is picking some of the letters from these vocabulary words, and it is supported by ICA-NL. And here, I would also mention the idea behind Vantage6 is implementation of personal health chain. The personal health chain is an analogy of a station representing the data source and a chain representing the research question that visiting the data station. In this way, the dataset is kept where it stays, and only the chain moves instead of sending the data to request. Also, with the links at the bottom, you can get more information on these two concepts or ideas. There are also other several components, for example, Fair Data Station, which Peter-Bram also mentioned in his slides. It's a metadata ingestion platform that helps users to record metadata. And yeah, also here we mentioned the SURF. And the SURF is the IT cooperative of Dutch education and the research institution. And we use the SURF Research Cloud. It's a service from SURF. Or essentially, it's a portal where you can easily build a virtual research environment. The last but not least component I would mention is the ISA abstract model. Since I think most of the audience are from life science, so I guess you may be familiar with this one. It's a specification for enabling discovering, exchange, and the publication of metadata from experiments in the life science. Okay, then with all this mentioned, then we can, again, I bring you this diagram, but there is a difference. We still see the stakeholders, dataset owner on the right and the dataset consumers on the left, but I replaced I provided the components which I mentioned in the previous slides here. So you see that dataset owner published the verified metadata to a metadata registry. So in our case, we're using fair data point, and also we provide a running fair data point instance with this URL. And behind the fair data point, as we mentioned, that we used the GraphDB to store the verified metadata. Then for the dataset consumer, can browse the dataset on the portal and also can perform semantic search query on the metadata. Specifically, this happens in the GraphDB. Then for an advanced user, the researcher, if the researcher would like to run an algorithm on a dataset, then with the help of the server, Ventisix server, he can send his computation request to the access controlled dataset. And the Ventisix server will interact with the Ventisix node, and also the Ventisix node will download a request from the Docker Hub, and the Ventisix node has access to the data storage or the dataset, and then the calculation will happen on the dataset, which also is on the administration of the dataset owner. Then after the calculation, the result will be returned back to the Ventisix server and then returned to the dataset consumer. So this workflow with the specific components in this diagram, I hope that it helps also present a more clear picture of the components of FDCube and how they interact with each other, also involving the different stakeholders. Then I would like to show you some short screens showing FDCube, its components, where you can find it. As we mentioned, that FDCube is developed and deployed on Surf Research Cloud. So here is a screenshot of the Surf Research Cloud, and FDCube currently already deployed as a catalog item. So that means a user, if you already have access to the Surf Research Cloud, then you can choose the FDCube by just clicking this item, then bring it up, then you will have the FDCube running. On this slide, I also show you that the Ventisix node and the Ventisix server also listed as a catalog item on Surf Research Cloud. Yeah, this all thanks for the contribution of from Surf, our colleague Yulia working on this. Then I would like to show you also some screenshots showing the process that happens. First is how to publish the metadata. So here we see this part of the diagram. We see the dataset owner. The dataset owner has some metadata, most case in a CSV format. And with FDCube, get a result. This metadata will be transformed into verified data and then published in this data catalog, data portal in our case, fed point. This is how it looks like. And we provide the script here on the GitHub with this link. And also we provided a Docker container, which also accessible with the GitHub link here. Then we continue with this second part of the diagram, the workflow for browsing the dataset portal or running a query. We zoom in for this part. The researcher which would like to browse the dataset, then researcher can view or browse this data portal. And also with the support of GraphDB, the researcher could run an semantic search or query in this GraphDB dialog, which user can run some SPARQL queries here. Then on the next slide, we zoom in the 26 part to see how a researcher can be helped if the researcher would like to run an algorithm on the dataset. 26 server provide a user interface, very friendly. Here you see that with setting up of the organization, collaboration, users and the nodes, all this etc. If all this setting up, then a user can create a new task. Here we show you a simple example that this algorithm is here. You see if you see my mouse, if you look at this, it's a URL pointing to a Docker hub, is where the 26 can host average algorithm, which is used to do a calculation on a data set. In our case, if you look at the right, this is our example data set. It's a CSV file with 2 columns, and our algorithm is going to do an average calculation on the age column. And then the result, the calculation is happened on the data set, and this result can be seen on this screenshot, which is on the 26 node. The result and will then be returned to the server and then be returned to the researcher. So this is the whole workflow for the researcher, helping the researcher to do algorithm on a data set. So with all this diagram and screenshot, I hope that it make it clear to you that how FDCube helps. So the main takeaway here is first, FDCube is an integrated cross-omic infrastructure that helps researchers in different stages of their research data life cycle. And until now, FDCube is deployed on Surf Research Cloud, and also it's possible to do a local deployment with the link on the GitHub. Okay, so that's my presentation. And here again, I show you this publication on the FDCube. And with this story, you can see more details there. Yeah, thanks for your time. And I think we still have time if anyone has questions, maybe can answer for this moment.
Yes, please. If you have any questions, you can add it to the Q&A session. Yes, there's a hand raised.
Yeah, Xiaofeng, great presentation. So I was just thinking from a scalability point of view, you have different components. You have a data catalog, you have a metadata repository process, and then a data analysis pipeline or like the usage with the Vantage server. So which one could we like scale up more the data catalog part of it for researchers? Could it be the data entry process, or so from what could you scale up for generic use for the researchers?
So if I understand you correctly, you are concerning about the scale up capability of FDCube.
Yeah.
So if you say scale up, then it means that large volume of data coming to need to be processed in that.
No, just from a usability point of view. So I think like data catalog part, like you have kind of a standardized data catalog where you even have a metadata repositories, like the metadata annotation process. And so I definitely think that, data catalog, like more and more researchers can use like the Fair Data Cube as a data catalog to find data, basically multi-omics data. So with like so, but I think if there's an ontology like gene ontology being a common point of query. So, but yeah, what's your like? What do you think researchers can actually use to kind of which component of FAIR they can really look into like from a larger extent to scale it up, basically. So
Yeah,
maybe within Radboud UMC or even within Netherlands in a larger extent.
I think Peter-Bram wants to say something on that.
Well, yeah, first of all, I think our vision was to reuse existing components as much as possible, as you've seen. The fair data point will be a central element also in the health RI infrastructure. And we, on the Radboud UMC fair data point, for example, published already multi-omics data that is then automatically also findable in the health RI catalog. So in a sense, that scaled up the findability of the data, because it's not only findable through the Fair Data Point Index, but now also through the Health RI catalog. And I hope that also in the European Open Science Cloud, this makes data findable. And also the other components are reused in other consortia. For example, the Vantage 6 is also kind of central component now to many federated infrastructures. So I think, yeah, that's part of the scalability to use components that are at least under full development within the Dutch and hopefully also international context.
Really nice. Very happy to see it grow.
And also add up to that, the 26, I think it supports the collaboration between multi-parties.
Yes, there are other questions in the Q&A window, and someone asked what parts of the metadata are generated using the FAIR data station, and is the FAIR data point configured to publish and display these?
Yeah, so in our case, to work, which we will see later on, the omics metadata we use in FAIR data station, and also other transcript that to make this transformation happens. Also, we mentioned the ISA metadata schema, which we used to annotate the metadata. And then the metadata being verified is published on the FAIR data point, which is public accessible and queryable. And I mentioned the Spark query, which machine can understand it.
And I guess there is the ShackWell configuration that's for the ISA metadata then, right?
Yeah, when publishing the metadata behind the FAIR points, there is a ShackWell regulation that to ensure the data quality, the metadata quality. For example, some mandatory fields or minimum count or maximum count of the metadata features, properties, etc.
Okay. Very nice.
Maybe
Thank you.
we should elaborate a little bit for the rest of the audience about ISA or ISA, investigation, study, essay kind of metadata, because that will not be familiar to everyone. But this is an established metadata scheme that, for example, also Metabolites uses, and it records metadata at different levels. So at the investigation level, at the study level, at the analysis level, and we try to adhere to those different levels. And indeed, a challenge was to get that properly published in the FAIR data point. But we have also made some efforts to realize that.
Yes, and Noruzi asked, how does the FAIR DataCube support data archiving, and for what duration?
Yeah, good question. So FAIR DataCube, in the diagram, we have the external storage there represented there. So FAIR DataCube would align with these external repositories, but for I would say for this moment we don't have much to present on that point. But we would like to align with, for example, we're using ISA as our metadata schema. So this ensures that we can easily align with some of the repository submitting requirements. And yeah, so that's the external storage. Then also there are some private one, for example, DRE. Yeah, we using DRE in Radboud UMC to store the private data set. And in the DRE, we install, we deploy a FAIR point there. So that's generally how FAIR DataCube supports the data archiving stage.
But in principle, any archiving solution is fine, as long as you have a DOI or stable URL pointing at that data source that is accessible. Yeah, there are some more questions about how to actually do this also locally. I think we should tackle that after Firda's presentation. And also, there is, she will announce a workshop where you can actually learn how to do it yourself. But maybe I can connect that also to the implementation in the DRE. So the question that was asked by Priyanka. So in principle, it's very well possible to install the FAIR DataCube within a DRE environment. There are some challenges, obviously, that you have to open the right ports in the digital research environment to expose the metadata that is supposed to be public, and also to allow incoming queries, for example, in the DRE environment. And there are efforts now going on to really also make sure that a DRE environment can be part of a personal health train data station or whatever, but this is still very much ongoing work. So it is possible to deploy it in the DRE, but you have to configure your DRE environment, and in particular, open the right ports to the outside world for this to take full advantage of the FAIR DataCube infrastructure.
Okay, I think we're going to take the rest of the questions after my presentation. And my name is Firdaus Badmos. I'm a data steward at the Radboud UMC at the CMBI department, and I've been working on the FAIR DataCube for almost a year now. And I'll be presenting the Trusted World of Corona to Work for Short project, which is a demonstration of the application of the FAIR DataCube to show FAIR data capabilities in the TWORK project. Yeah, so the TWORK project started as a response to the influx of data during the COVID-19 pandemic. The pandemic led to a rapid increase in research publications and data sets, creating an overwhelming knowledge pool of information for researchers. And many of these findings were sometimes false and non-reproducible, with over 370,000 articles in 8,000 journals over the period of 3 years. It was essential to find a way to keep this data fair. Applying fair data capabilities to such knowledge pool is essential for the management and access to research results. It is also crucial for the public policy process and for making economic decisions. The fair data capabilities also enables collaborative research by allowing the formulation and testing of hypotheses. It also allows the annotation of associations with trustworthy opinions, ultimately providing a rich source of metadata for future research. So what was the process of enabling the fair data capabilities for this work project? We started by getting our data set. The data set contained 241 COVID-19 diagnosed patients with 258 healthy controls. And this data came from 2 independent studies. We had samples with transcriptomics, proteomics, and metabolomics measurements. We also had phenotype and targeted lab measurement data. And this data covered 3 groups of patients: the COVID-19 patients in ICU, COVID-19 patients not in ICU, and healthy control. Then we proceeded to the process of verification and onboarding. And just like Xiaofeng mentioned earlier, this process starts with a researcher that onboards the data set and the metadata on a fair data point. And behind the fair data points, we have the knowledge graph where the data is actually stored and queryable, and the fair data points client where the metadata is displayed and findable. At the back end, like Xiaofeng mentioned earlier, we have the ISA data structure. And for sensitive data, for example, a researcher can also send an algorithm to a data set that has been discovered if the owner of the data set does not want to share the data or if it contains sensitive information. So this is how we use the 3 components of the fair data cube for these data sets. And this is an example of what the data sets looks like before verification. This data is publicly available and is not sensitive, and these are just some of the subjects and their COVID-19 status data. After verification, the data looks more like this, and this is a total file containing triples. And a triple it's made up of a subject. This is a subject, the object, and the predicate. The subject is the entity being described. Here, this is a subject or an individual. The object is the value or the entity linked to the subject. In this case, it's the COVID-19 status. And the predicate describes the relationship between the subject and object. Here, it shows the Boolean yes. So this is just one of the examples of what data looks like after we have verified the data. This is another example of the verified data where the Kirby identifiers of the metabolites in the metabolomics data is displayed. And as we know, metabolomics data can be difficult to match because of varying database identifier format. And like a lot of people mentioned during the poll, this is one of the biggest issues that people face when doing multi-omics data. And in the subsequent slides, I'm going to show in more details how we matched these identifiers. And this kind of process of verification is one of the ways that the Fair Data Cube can be used to facilitate the integration of data from different sources and omics layers. After the data sets had been verified and transformed, we onboarded the metadata to the fair data points, and this is what it looks like. On the left, we see a description of the Trusted World of Corona project. So if you go to the CMBI Fair Data Point and you search for the TWORK project, this is what you see. And these are some of the metadata that is present in the data set that can also be visible and findable by someone interested in this study. So now that we have the Fair Data Point set up and the metadata is on the Fair Data Point, we might want to search for studies that contain COVID and multi-omics in their title. So this is the triple store. This is the graph DB that is connected to the Fair Data Points. And we do this by running a query. For example, we are selecting 3 values, and we did a regex filter for COVID-19 and multi-omics and limited the result to 1,000. So basically, we are looking for studies that have COVID-19 and multi-omics in their titles, for example. And this is an example of what the result looks like, and it returned a study ID, the name of the study. So this is one of the ways that you can use Sparkle queries to discover data. Another kind of query that can be run is the one displayed here, to query the triple store to know the COVID-19 status and the ICU status of the subject. This query starts by defining the prefix XSD, and then it's select the 3 components that we are interested in discovering, which is the individual, the subject, the COVID-19 status, and also the ICU status. And we filtered for COVID equals to yes to be sure that we only get results of COVID-19 positive patients. And here is how the result is displayed. So these are some of the ways that a data set that has been onboarded on the Fair Data Point can be queried through the graph database. Moving on to the multi-omics data analysis of the data, we know that in multi-omics data, some of the challenges we face is that data preparation can be very time consuming and energy consuming. And sometimes we deal with sensitive data that cannot be shared. So we use the FDCube to create a workflow that does this securely and also saves energy and time. The goal of this analysis was to integrate different omics layers in a data set, and to also integrate pathway data. So we are bringing together 2 data sources for analysis. The general workflow we used started by querying to find studies on COVID-19, which I showed a bit earlier, and also querying a pathway database to get pathway data for a specific pathway we're interested in studying. The data we got from the GraphDB contained omics data, 2 omics data, the proteomics and the transcriptomics data for the 3 patient groups. And these 2 data sources were integrated with BridgeDB. I'm going to explain a little bit more on BridgeDB soon, which we then analyzed. I mentioned that we used data from pathway data, and we used WikiPathways, which is a FAIR database that contains biological pathway information on biological. We know that these biological pathways give an intuitive representation of biological processes. So that is why we decided to integrate pathway data. And this database is a FAIR data source that also has a Sparkle endpoints that can be queried. We specifically looked at the SARS-CoV-2 innate immunity evasion and self-specific immune response pathway for this analysis. I mentioned BridgeDB earlier, and to connect the different omics layers and the pathway data, we used BridgeDB. And BridgeDB is a framework of map that is used to map identifiers from proteomics data and transcriptomics data and different types of biological databases. And like I said earlier, having different identifiers is one of the biggest problems in multi-omics analysis. However, BridgeDB creates a service for mapping identifiers, notwithstanding whatever database source they came from. To show the workflow in more detail, I'm going to go through a couple of images. So first, we started by querying to find studies on COVID-19 with multi-omics data. Then we fetched the data of the individuals included in the study, including their COVID-19 and ICU admission status. After that, we got the subject identifiers for each subject and used this to fetch the samples collected from the subject. Eventually, we had the proteomics data and the transcriptomics data. Moving on to the pathway data, we ran a keyword search query on wiki pathways. Then we queried the pathway that we're interested in, which was the SARS-CoV-2 immunity pathway. Eventually, we extracted the gene products of this specific pathway. Then we got the identifiers of the transcriptomics features, and then we combined this using the fetch mapping script. The fetch mapping script used BridgeDB. So what we did is that we looked at the transcriptomics features and compared them to the gene products from wiki pathways and see if there are any common elements. And the way we find these common elements is to use BridgeDB to convert each, for example, each transcriptomics features to all the possible database identifiers that they can have so that we can find a match in the wiki pathways data. I'm going to explain a little bit more in the following slides. So we repeated the process for the proteomics data. We got the identifiers and we matched them to the wiki pathways data. Eventually, we merged these different omics features and then we analyzed them to see their different levels. Like I mentioned earlier, we're able to combine data from two fair sources for this analysis. In the next couple of slides, I'm going to show a Jupyter notebook of how we did this process and this process that I've circled in red. So we start by importing pandas and then run the keyword search query module. This module takes the endpoint URL of Sparkle of wiki pathways and the SARS-CoV-2 keyword, which is the keyword that we are interested in finding pathways on. This result returns a couple of results, and we were interested in this last pathway with the WP5039 identifier. So we ran the fetch gene products module, specifying the identifier we are interested in and an output file. When we run this, we get a message that the gene product for this pathway has been saved. And when we inspect the file, this is what we see. So these are different identifiers for the gene products in the specific pathway. After that, we move on to the proteomics data, and we run the get features identifiers module to extract the identifiers of the proteomics data. This is what the results looks like after the extraction. And then we move on to fetch the mappings. That is, look for the proteomics identifiers that are present in the pathway gene products that we just extracted. And this is done by running the fetch mappings. Here we can see that we specified the source, basically the source database. And this is necessary for BridgeDB to map these database identifiers. So every possible or all the synonyms of the identifiers in different databases to be able to compare this file to the Wikipathway gene products file. Eventually, we ended up with 3 proteins present in the gene products of the pathway of interest. We repeated this process for the transcriptomics data. First, we got the identifiers. The identifiers are displayed here. Then we ran the fetch mappings module to get these identifiers that are present in the gene products from Wikipathways. These are some of the proteomics data that we found in the pathway of interest. Eventually, we combined the proteomics and the transcriptomics results to find any overlaps. And we ended up with 3 features. Then we plotted the features to show the different levels across different patient groups. This is what the results looked like after we had combined the proteomics features and the transcriptomics features. Like I showed earlier, these were the 3 features that we found to be common to both the proteins and the transcripts for this data. And this is what the different levels of the transcripts looks like for the different patient groups. And for the protein levels, we can see that there's a difference between the COVID-19 non-ICU patients and COVID-19 ICU patients and their IL-10 protein level. We just plotted the first feature. It's also possible to show the different levels of the other features that we found. So in summary, this workflow shows how the FDCube provides tooling for verification and integration of different levels of omics data. And we can see how it makes the multi-omics analysis efficient. And although we created this FDCube for multi-omics data analysis, it can also be expanded and used for other multimodal data. Yeah, these are some of the links to the tools and data sets. And yeah, we have published a paper on the FDCube, and it can be accessed through this DOI. Thank you. Any questions?
Yeah, I see at least one question that was not asked before. Just wondering, is there a specific reason why you do not use the Sparkle query capability of the FAIR data point, and is that too limited?
Yes, I think the FAIR data point is constantly being developed and improved, and we hope that this would be possible in the nearest future. But right now, this is the way we can currently query the data. It has to be connected to a triple stop. Yeah.
Mm-hmm.
Okay. Any other questions?
Yeah. Maybe add to that question. If I understand correctly, mentioned the triple store behind the FAIR point.
Mm-hmm.
Yeah. Yeah, the triple store can be, it has provide several options. So in our case, we use GraphDB.
Yes.
Yeah, maybe then it's time to go back to the questions that were asked earlier. So first, a question by Kumar Singh. Could you explain briefly how could I use FAIR DataCube locally, or for example, with non-human data?
Maybe I can answer the first part of the question to using FAIR DataCube locally, and then regarding the human data, maybe pick up by one of you. Yeah. So yeah, currently what we show is that we deployed the FAIR DataCube on Surf Research Cloud cloud deployment, which was quite easy. If you have access to the Surf Research Cloud, you can easily bring it up by picking from the catalog, use FAIR DataCube and also Vendor 6 server and the node, and all this code, etc., are also public on the GitHub, and we will soon we will make the local deployment easily. Yeah. we'll keep you updated, and will be available in a hands-on workshop.
Yes, we are planning to have a hands-on workshop where we can show how the FDCube can be deployed on a local system and also can be deployed using the Stuff Research Cloud. So yes, we are planning a workshop for that.
Yeah, maybe when you use non-human data, so are working with non-human data that are not sensitive, and then you can also decide to implement or deploy parts of the FDCube because you're not likely to use the Advantage X component then, for example. And since the system is entirely modular, it is also easy to do that and only install the components that you need. Then the second question by Surya Gupta is, how can we take advantage of this pipeline in-house to store, process our own in-house dataset?
If I understand the question correctly, you want to know how you can use this pipeline to store and process your own data. On the GitHub, like Xiaofeng mentioned, we have the scripts for the different processes that we followed to verify this data and to also onboard the data set to the fair data points. If you want to use the fair data station, there's also, we could also provide a link on how to use the different metadata sheet and choose, for example, what kind of data sets you want to onboard. And there you can create your own metadata sheets that can then be used to onboard the metadata on the fair data point. I think we can also show this during the workshop, because we are going to be showing how to onboard an example data set on the fair data point, I believe. If
Surya, does this answer your question, or do you have further questions? You can also use the microphone to just intervene.
Yeah, thank you so much. I actually wanted to ask that, okay, if I want to follow the architect at the moment that you have used to actually deploy all the data, to process it, and to then following with that to query the data for the further analysis. I do have data in the house that we have a lot of data that we produce, and not all of the data can be actually submitted online, but I would like everybody to use that data. Bit similar what you did, making that a fair data, but in-house fair data first, and then of course we can go with next step for the public one. But I want to set up the same system, same architect, so that all the students from different omics can actually cross-talk with the data and actually query the data for their own scientific understanding. And this is something that's really interesting for me, the architect, and I really like it. The one that I'm trying to follow at the moment is using the cloud architect, so that that we can actually query the data using SparkQL or XQL, depends on that. But I think this is more interesting. But the thing that the question that I have following what I wrote before is that if I do want to use the architect, what part of it can I just use as locally? So like I can install the same architect on the servers locally, but can it be locally, or it has to be partly online, depending on the algorithm or the tools that you used?
So maybe let me quickly respond. In terms of locally, the FDCube, the whole FDCube, including FedPoint, GraphDB, 26 server, and the node, all these things can be locally.
Okay. Okay, and is there a way whom I can contact if I would like to get a help from this? Because of course I'm not an expert. I know graph database, I know every other things, but how to do it together, I would really like to talk to somebody and collaborate on that.
Yeah, you can reach out. Maybe reach out by email or on the GitHub issue via GitHub issues.
Okay, so you will share the link for the GitHub?
Yes, the link to the GitHub is in the presentations, and we would share the presentations, but I could also put the link to the GitHub in the chat, and you could also reach out to me. I'll share my email at the end of this session, and you can always send me an email.
Okay, that sounds good. Thank you so much.
Yeah, you can also use the general Cross-Omics email address, so to where you can get support on all these kind of issues. And of course, we will announce the workshop where I think this would be the first step now for people who are interested in deploying things locally or already on a cloud. And I think that will be a first step, and then you can get further support in this after that.
Okay, looking forward for the workshop then.
Before we then announce that, I have a few more polls for you that may also raise further discussion. So let's first go to this one. So this is basically to check whether the FAIR Data Cube meets your needs. So do you think that a FAIR Data Cube can facilitate your data analysis? Hear your opinions on that. 17 responses so far. Maybe others also want to cast their votes. And then perhaps the people who think it is still questionable whether the FAIR Data Cube can facilitate their data analysis, maybe you can just raise your hand if you want to comment on this, and what are things you would need actually for the FAIR Data Cube to deliver to make it more useful? Who can I give the floor on that to? Who wants to raise their hand or put something in the chat? It's also possible. Anyone who can comment on what they are missing or what they expect extra from the FAIR Data Cube? Don't be shy. This is your chance to add extra functionality, maybe. Yeah. Simon Daniel is commenting. So Simon comments, the problem for our situation is not so much the cube itself, it's that we have multiple partners who would need to each be convinced to collectively adopt the system. Yeah. Well, I think that is a good point. So we have one example, project, big infrastructure project where, or at least two examples at the moment, who want to adopt the FAIR Data Cube infrastructure. But indeed, it makes sense that if you have collaborative partners, that you all use a system that is at least compatible with respect to the accessibility through FAIR data points, and on also to have some agreement on the metadata schemas that you use. Yeah, I think the convincing part is perhaps that yeah, you can make life easier also for your partners, and that you do not have to copy the data between multiple partners, but in principle could work on this in a federated setting where the data stays as much as possible with the different partners. But yeah, I agree. It needs some convincing, and it helps to at least well have a common vision on how you would like to integrate the partners, the different data sets from multiple partners. If there's anything we can help you with, Simon, let us also know. Okay, and the last poll I would have for you is actually on the workshop, but maybe Firdaus, maybe you first start the announcement, and then we can launch the poll after that.
Yes. So yes, we are planning to have the hands-on workshop on the 9th of April at the staff location in Utrecht, would be sending out the invitation to everyone who attended this webinar. So if you are interested to learn more on how to set this up locally or using the soft research infrastructure, you're welcome to join us on the 9th of April in Utrecht. And if you have any other questions or or comments, you can reach out to me at the email shown on the screen. We'll also be sending out the presentations from this webinar to everyone, so you can access the links and the resources that we have shown today. Yeah, thank you.
And then we have 2 last questions for you. So this is the first one. So just to test interest for the hands-on workshop. So how many of you would be interested to attend the hands-on workshop? 16 responses so far. Okay, so there's at least a high number of you who would be interested to learn more and get some hands-on experience. So thank you for that. And then maybe as a kind of question to test what your current level of understanding of the FAIR Data Cube is and what we can actually achieve after the workshop is this last question. Do you know what the FAIR Data Cube can do for you after this webinar? And maybe if you want to know specific more things, learn about those in the workshop, then you can also put those in the chat. So from what I conclude from this poll is that it was kind of an introduction on the FAIR Data Cube, but it was not completely clear yet. So there is definitely a need to continue this, and hopefully we will then be able to meet up with many of you during the workshop on April 9. And with that, when there are no further questions, Firdaus, I think we can perhaps close.
Yes, thank you again for joining us today, and please do not hesitate to reach out if you have any other questions or suggestions, or even things that you would like to see in the workshop. Yeah, thank you for joining.
Okay, thank you.
Thank you.
Bye.
Bye-bye.